So we have this idea of transgenic biotechnology, which is, as I said in the previous lecture, basically just taking the genome of one organism and putting part of it into another organism. And so an example of this is HGH. It's usually secreted by the pituitary. Some people are missing this, and so you need to, hopefully you can get it to them when they're very young. And we used to find people that were recently deceased and extracted it from them. And this was pretty costly, and it turns out not particularly safe for a number of reasons. But now we take the human gene for HGH. Remember from the last section that a gene is a segment of DNA that codes for a particular protein, and HGH is a protein. We took the gene for HGH, we put it into an E. coli, and we said, okay, E. coli, go, make us a bunch of human growth hormone. And so now we're able to have these little factories that we're able to just take whatever gene we want, more or less, put it into them and have them make it for us. So there's some tools that you need in order to be able to undergo these various forms of biotechnology, one of which is restriction enzymes. And restriction enzymes can basically just be thought of as little molecular scissors. They're going to cut at a very specific portion, or very specific location in your DNA. And these things are made by bacteria, and bacteria evolved them so that they could defend against viruses. So if you have viral DNA coming in, you're a bacteria, you certainly don't want that, right? So you unleash your restriction enzymes, and they're going to cut the viral DNA into pieces. Now, these restriction enzymes are typically very specific. They only cut at particular locations, which are called restriction sequences. And those restriction sequences are going to be found in different spots in your genome. And in a second, we're going to talk about plasmids, and hopefully this will make a little bit more sense why we're using these tools. So the restriction enzymes leave sticky ends that you can use to paste things back in, like DNA Velcro. Okay, and this is useful for taking a piece of human DNA and sticking it into a piece of bacterial DNA. All right, so restriction enzymes are like molecular scissors. You can use them to cut pieces of DNA, and then what you can do is take another piece of DNA and paste it in. You can take another piece of DNA and paste it in. Different restriction enzymes each have their own recognition site, so different restriction enzymes are going to cut at different places, so you need different scissors to make different cuts. And there are over 1,000 of these restriction enzymes that we've characterized that we know about. So this is just a picture of what I was talking about a moment ago. You have your double helix that's kind of been flattened out here for us to be able to see more easily. And then highlighted in white here, you have your restriction sites. So remember, these restriction enzymes are going to cut at very specific places along the genome. So they're going to come in here, and it's going to cut at this restriction site. It's also going to cut at this restriction site, which leaves you with a DNA fragment. And so you can do a few things here. Let me get my pointer. You can either take this piece of DNA and you can utilize it for whatever you want. Maybe you're interested in whatever piece of the genome is here. Or you can take these leftover bits and paste your own DNA fragment in. You can paste whatever piece of DNA you want into there. And that's really cool and really fantastic, and has allowed us to do a ton of stuff in biotechnology in the last 30 or so years. So the next idea that we're going to talk about here is plasmids. Plasmids help us to put DNA into bacteria. Okay? Plasmids help us to put DNA into bacteria. So you'll remember that bacteria are prokaryotes. They don't have a nucleus, but they still have DNA inside of their cells. They still have DNA. And they have a chromosome in there. And most of their genetic information comes from those chromosomes. But what someone discovered is that they also occasionally have these things called plasmids. Plasmids are these pieces of DNA that are not part of the chromosome that the bacteria can use to do different stuff. And they can also be exchanged from one bacteria to the next. So you can take a plasmid from one bacteria and you can put it into another bacteria. So And these plasmids contain certain genes. So let's say you have a gene that's going to make, I don't know, this is dumb, but let's say you have a gene that's going to make a bacteria turn purple, and you have it on a plasmid. You could just take that plasmid and put it into another bacteria, and guess what? That bacteria is going to be turned purple. Okay? And like I said, these things are able, this is a great way for bacteria to be able to exchange genetic information, because bacteria don't have sex. They will occasionally exchange bacteria in what appears to be a slightly sexual reproduction, but most of the time they're reproducing via binary fission, and they're picking up these different plasmids and exchanging these plasmids with one another so that they can do different stuff that maybe they couldn't have done before. So here's our bacterial chromosome inside our bacteria. There's no nucleus or anything like that, but you'll also notice this plasmid here. And like I was saying earlier, plasmids can do all sorts of different things. Maybe this plasmid is giving this bacteria resistance to some antibiotic, or maybe it's allowing this plasmid or allowing this bacteria to live in a really, really cold place because it has certain proteins that are able to compensate for the cold. So getting human genes into plasmids is kind of the next topic here. So what you do is you take your human DNA, you cut it with a restriction enzyme, and you take your plasmid and you cut your plasmid open with a restriction enzyme. Okay? Then you take your piece of human DNA, you put it with the plasmid, you put certain enzymes in there, and that causes your human DNA to now be permanently attached to that plasmid. Once those pieces of DNA are connected, you have what is referred to as recombinant DNA. So that's an important word to know. Once you have a piece of DNA that's made from different organisms, it's called recombinant DNA. There are different vehicles that can help transfer DNA into a cell, which are called cloning vectors, which are very often plasmid-based, and maybe we'll talk about those later. So here's a picture of what I was just talking about. You have a human protein, this little green triangle-looking thing. I don't know what it is. Maybe it's HGH, maybe it's insulin, maybe it's something else. It's not super pivotal for this, but you know where the gene is that makes this protein. You know where it is. You've sequenced the genome. You've studied it enough. You figured it out. You know where the gene is on the chromosome. So you cut it out. You take it and you're going to remove it from the rest of the DNA. And the way you do that, you guys remember the way you do that? You use restriction enzymes. You take and cut it out with a restriction enzyme, and you take your plasmid, you grab that out of a bacteria. You're not trying to get the chromosome, you're trying to get the plasmid. So you take the plasmid out, and you also cut that. You cut that with a restriction enzyme. Okay? That process, or that is called recombinant DNA. You might also call it a ligation, but you don't need to know that. Then, after you have your piece of recombinant DNA that's a little bit human, that's a little bit bacteria, you do what's called transformation. You take that plasmid and you put it back into the bacteria. You don't need to know this, but very often the way that you're going to do that is using electrophoresis, or you're going to put electricity into it and force the plasmid into the bacteria, because bacteria will take up plasmids, but it can be kind of rare, and if it's not a plasmid that they particularly want or like, they won't take it up, but you can kind of force them to get the plasmid. And then you have all these bacterial colonies that have your protein or your gene of interest on them, and they start producing that gene for them. And there are different tools and different tricks you can use to force the bacteria to start making the protein for them. But this is a really, really fascinating and super cool process. Okay? We are making human stuff with bacteria. I don't know, it blows my mind. So, like I just described, transformation is the process of taking your plasmid that you have created, also called a cloning vector, and putting it into the bacteria that you want. So you can take a plasmid that you haven't messed with. You just have a gene that you're interested in, maybe, and you want to put it into another bacteria. You can do that, but you can also using those restriction enzymes, cut your own DNA into a plasmid, or cut the DNA that you're interested in into a plasmid and have it start making that protein for you. All right? This is called transformation. So there are already over 150 estimated biotechnology drugs and vaccines approved by the FDA. This is old data. I'm sure there are more than that now. Goats, yeast, hamster cells, all bioreactors that we use to make different things. I talked about this earlier, but goats are used to produce a human protein that helps with blood clotting. You can take a goat, and it's a different process than a bacterial transformation because goats are much more complicated. Animals are much more complicated, but you can make them produce these things for you using biotechnology. We also have genetically modified crops. So you have things like these rice grains, which are basically used to make extra beta carotene to help people who might be starving without it or might be lacking this particular vitamin. And you can actually take it, put it into the plant, have the plant start making it for you, and get them the vitamin A that they need.